Good morning. I think it's time to get started. Good afternoon, good morning, everyone, depending on where you are. I'm Adil Sariwi, happy to be the moderator of this session of the webinar series hosted by INRA Morocco to discuss trends and innovations in agriculture. I'd like to welcome all the participants from different countries for taking the time to attend this webinar, and I'm looking forward to their potential inputs and comments in the QA session. Before going further in this session, I would like to ask you all to please mention your full names and affiliations on the participant lists. Participants who cannot be identified will be logged off in 5 minutes. Please also make sure your microphones are off throughout the session, unless you are given the floor to speak. Thanks for your understanding. Today, we are delivering the 5th session of the 6-webinar mini-series that started in November last year, focusing on hemp or cannabis. This series of webinars is framed by the promulgation of the Law 1320 on the legal use of cannabis in Morocco. This is a new bill that marks a turning point in the use and perception of these plants and its outlets in our country. It opens several perspectives and imposes challenges to current and future potential actors working in the related value chains. Therefore, there is a growing need in Morocco to exchange scientific knowledge and learn from practical experiences to fully benefit the Moroccan economy for the opportunities offered by the new law. 4 speakers have taken part so far to this webinar series. They spanned a wide array of topics in their talks, ranging from history and legislations to therapeutic use of cannabis. Continuing along this path, I am delighted to introduce our today's speaker, Dr. KARLA Gage, Associate Professor of Weed Science and Plant Biology in the Schools of Agricultural Sciences and Biological Sciences at Southern Illinois University, Carbondale. Dr. Gage completed bachelor and master degrees in biology at the University of Memphis and a PhD in plant biology with a focus on weed ecology at the University of Illinois. Dr. Gage joined the faculty of the same university in 2015 and enjoys teaching and research in applied management of agricultural systems. Dr. Gage's research focuses on weed control in Midwestern U.S. cropping systems in the rotational crops of corn, soybean, wheat, and sorghum, and now, perhaps more importantly for us, industrial hemp. The Gage Lab focuses on the use of integrated weed management to slow the evolution of herbicide resistance and suppress weeds. Dr. Gage has graduated 10 master's students and currently advises or co-advises 9 master's students and 3 PhD students. She has also mentored dozens undergraduates, researchers, and students and published 22 papers and 3 book chapters. In addition, she has authors or co-authored the 69 conference proceedings and served as a reviewer for several journals and as associate editor for Frontiers in Agronomy. She lives in Carbondale, Illinois with her husband and lovely 12-year-old twins and enjoys hiking and botanizing in the Shawnee National Forest. Dr. Gage will share with us her insights through a talk entitled Growing Hemp, Cannabis Sativa for Fiber and Seed in the Midwestern USA, Challenges, Lessons, and Opportunities in Cultivation. The main talk will take up to 35-40 minutes, and we will have a QA session that will last around 20 minutes. Please post your questions on the chat dashboard As the presentation progresses, participants will also be given the chance to ask questions or provide comments orally by raising their electronic hands during the Q&A sessions. Questions can be asked in French or English. Dr. Gage, we are looking forward to listening to you and knowing more about your exciting topic. The floor is yours.
Thank you so much for that lovely introduction. I'm so happy to be here with you all today. I'll give you some of the background as to how I got involved in cannabis research, and I'll tell you kind of where my lab is going with that. So first of all, I would love to thank everyone who is associated with the AMALS conference. Conference organizers Khalid Meksum and Majid Abdel introduced me to so many wonderful people now, and I'm really thankful for all the connections that I've made with Moroccan researchers. So I really appreciate the opportunity to be here to meet with you all again. So this is where I'm located. When I say Midwestern US, I am in the southernmost part of Illinois in the Midwest. So you may be familiar with Chicago. Chicago is also in Illinois, but it's very far north. So we are very rural. There's a lot of forested land, a lot of natural areas around us where we are. And this is an image of our university farms where I do research. And my research has typically been focused around corn and soybean production. So that's the predominant rotation in Midwestern agriculture. Those two crops rotate. It's a very simplified rotation. And some growers may plant winter wheat after corn going into soybeans, and some growers may plant grain sorghum instead of corn in some of those rotational years. But hemp is now potentially a new rotational crop for Midwestern production. There are issues in market development, meaning that if you grow hemp right now, you're limited in the places that you're able to sell it. But that is slowly changing over time as we have more fiber processors coming online and accepting product. So this is something that's still evolving. I am a weed ecologist. I've studied ecology for decades, and now I get to apply that knowledge in agricultural systems. But I'm very interested in the interactions or the relationships between weeds in particular and the crops and other weeds. So all the biotic factors that influence weed growth and development. Those are also interactions with insects and pathogens and pollinators. So all of those things, those ecological interactions can be utilized in improving agricultural production. So this is basically the science that investigates how ecology can be applied to agricultural systems in an effort to improve production. So I want to just briefly go through a history of hemp in the U.S. I really enjoyed listening to some of the Moroccan scientists present the history of cannabis in Morocco. So I thought I would do the same. And then I'm briefly going to talk about the legalization of hemp and cannabis in the US as an overview. And then there are three main commodity types. So if you're growing cannabis or hemp, you can produce three commodities. And then I want to go through some challenges and what we've learned and what we need moving forward. And then lastly, from my perspective as a weed ecologist, I want to talk about the opportunities for hemp and how that may improve agronomic production. So the definition of hemp was defined in the 2018 Farm Bill. This piece of legislation allowed growers in the US to cultivate hemp with within a certain framework of rules. But this distinguishes hemp and marijuana, and I have this term highlighted for a reason. But hemp, this is defined as the plant cannabis sativa and any part of that plant, including the seeds, all derivatives, extracts, cannabinoids, etc., whether growing or not, with a delta-9 THC concentration of not more than 0.3% on a dry weight basis. So this is an arbitrary cutoff. This 0.3% THC limit was set a long time ago by researchers Small and Cronquist, and they were looking for a way to separate hemp from high THC cannabis in their taxonomic studies. And it became, because of this one publication, it became the threshold for defining hemp versus high THC cannabis. So this is arbitrary, and in the future, a lot of researchers and, well, a lot of people in the US would like to see this increased to a 1% THC on a dry weight basis. So I don't like the term marijuana. There's an issue with terminology for plants that are greater than 0.3% THC. The term marijuana is actually of discriminatory origins, and it's something that we should acknowledge and move past, I believe. But you'll find the term in legal writings within the U.S. to define high THC cannabis, and it's a very common term to refer to high THC cannabis. But it's associated with anti-immigration sentiment, and it became associated with crime as well throughout our history. But if you refer to the high THC plants as cannabis, which some people do, cannabis is the genus of the plant. So when I say cannabis, I'm thinking industrial hemp as well, but that's not the case for a lot of others, especially again in the legal world, right? Because cannabis has now also become synonymous with high THC plants. So we have a little bit of work to do as far as our terminology, I believe, to move past these issues. So just a very brief history, I think it's important to acknowledge the original habitat of cannabis, just because that kind of tells us what the needs of the plant are now as far as cultivation. But prior to human influence, cannabis was found in mammalian manured soil. So cannabis was associated with large mammalian grazers at one time, and it needs continually moist yet well-drained soils. So alluvial systems near rivers were ideal for the establishment of this plant. And those alluvial areas also had frequent disturbance, and the plant also needs frequent disturbance for establishment. So cannabis is evolutionarily older than humans, and it would have evolved in the absence of humans. But there has been significant coevolution since humans began to recognize the benefits of cannabis. And there's this rubbish heap hypothesis that cannabis actually evolved with human campsites. So as humans began to recognize the medicinal benefits and the benefits of the plant as far as fiber and all of its uses, they began to cultivate it, and it was found in rubbish heaps within these settlements, and that's how coevolutions began. So in the 1800s and early 1900s, several states within the U.S. grew hemp, cannabis, and you can see the potential distribution map here. And again, this is where I am with the red star here. It was grown in Illinois, all across Illinois. By 1929, it was marginally or not profitable for growers, though. Cotton began to come in and replace hemp in utility, so it became not feasible for growers. However, the production of hemp was influenced by war, and in World War II, there was a campaign, a propaganda campaign, to encourage growers to produce hemp, and you see the Grow Hemp for the War poster here. So those legacy hemp stands, there's still hemp populations that exist from this time period in Illinois, and it's referred to as victory hemp because of these propaganda campaigns. So if you're growing hemp in the World War II era or before, it would have looked something like this. So it would have been planted in very dense stands for fiber. It would have been hand harvested and then either laid on the ground to ret, so that the fibers separate from the stem, or it would have been put in these shocks that you see in the back for uniform retting as well. So that's where microbial action comes in and begins to sort of degrade the hemp stalk. And those are just some more pictures of what that might have looked like. So heavily labor intensive at this time. And then after retting, after the fibers were loosened from the stem through microbial action, they would have been run through one of these handheld implements to break the stalks and release the fiber from the stem for further processing. So in the 1960s and 1970s, well, there was a period of prohibition that began in the 30s, and it was temporarily relaxed with the anti-war movement of the Vietnam War. And then President Richard Nixon and Congress passed the Controlled Substances Act in 1970. And this began this most recent period of prohibition that we're now coming out of, where cannabis, heroin, and LSD were considered Schedule I narcotics and illegal at the federal level. And this Schedule I means that there's no medical benefit and the highest potential for abuse. So we know that that's not true of cannabis. And there's been movement to remove cannabis from the Schedule I narcotic list. But basically in 1972, there was this war on drugs that was established. And there was a report from Congress that signaled that cannabis was not a threat, but that was rejected by President Nixon at the time. And then the DEA was created in 1974. There was the Anti-Drug Abuse Act in 1986, where the possession of 100 cannabis plants was equivalent to 100 grams of heroin in the sentence. And then later this was amended to 3 strikes and you're out, and you could get a life sentence if you were caught with cannabis 3 times. There were 400,000 inmates convicted for cannabis in 1986, but 1.5 million in 2015. So marijuana at this time was about 50% of all drug arrests. One of the also unfortunate things that happened was the destruction of the seed collections by the U.S. Department of Agriculture and the refusal of new seed collections. So you can see the variety of seed size here. This is a farrow plant from the Kashmir region. And these are culinary seeds from China that you see there. So the fact that we lost this repository for genetic diversity was very unfortunate for us today as we look for new materials to breed with. So the recent legalization, just very quickly, we're coming out of this era of prohibition here that you see from 1900 to the 1980s. There was a landmark case in 2014 where the Farm Bill allowed researchers to do research on cannabis now, hemp in particular, so the low THC plants. So it became possible for university researchers to work on it at that time. But in 2018, the Farm Bill legalized hemp for cultivation by growers. And since then, there's been a little bit more. In 2021, this Marijuana Opportunity Reinvestment and Expungement Act passed the House and is now hopefully going to be taken up by the Senate. If this happens, that will allow the federal legalization of cannabis. So that will expunge all previous convictions and allow us to move forward. It will take it off the Schedule I narcotics list as well. And then the projection is that by 2025, the cannabis market will be $41.5 billion in the US. So there's a lot of opportunity for economic growth here. So as far as production, I just want to go through some of the differences in production for these commodity types. So starting with the CBD type cannabis, this would be very similar to what a high THC plant would look like. But these plants would be very low in THC levels and cultivated for CBD extraction. So they're plants of medium height with lots of branches, and they're planted at very low densities. So there'll be one plant with a lot of space between it and its neighbors. And this would be typical of a CBD production field. Now, the plastic that you see here would be laid down for weed control so that manual weeding doesn't have to happen over such a large acreage. And this is going to be a little different. The plants look very similar to high THC plants, but typically high THC medical or recreational cannabis would be grown indoors or at least in a greenhouse because of the phytosanitary controls that are needed for the medical production. So this would be what a high CBD plant or a medical plant would look like. And then there's a close-up picture of the trichomes of the plant. And these trichomes are plant hairs. They have glands on the tips of them, and that's where you'll find the terpenes and the cannabinoids in high concentration. So that's That's the product of this plant, is the trichomes, and harvesting the plant for the compounds that are found in the trichomes. So a large number of those trichomes are found on these paragonal bracts. So you see the bract here around the plant ovary. So this is a single flower, and you see the trichomes on the bract. If you remove the bract, you can see the ovary, and this would be a fertilized ovule here covered in a translucent perianth. So here, if you look at the picture on the top right, you see the perianth within that bract that is covering about 2/3 of the seed. And anatomically, this is very interesting because the perianth is equivalent to the petals and the sepal in a typical angiosperm. So that's interesting as far as anatomy. So typically CBD plants would be started in a greenhouse from seed or cuttings, and they would be transplanted into the field. And if growers are working across a large acreage, they probably have some sort of machinery to help them transplant. And this is just an example of a transplanter where individuals are dropping transplants into into this carousel, and it's being planted as they move across the field. There are issues when you use cuttings for propagation in the field. Cannabis has a tendency to go root bound if it's left in a small pot for too long. And this is an example of a root bound plant where the roots just keep growing around in a circle. And if you transplant those plants without opening the root system and allowing it to grow outward from the plant, then that can actually girdle the plant itself. So the root system can limit the plant's growth. And we do see this, at least we did see this early on in CBD production in the US. So a grower of any type of cannabis has to obtain a certificate of analysis. They have to put in a harvest notification 4 weeks in advance of harvest, and they have to obtain this certificate that moves along with the product throughout all of the processing afterwards. So this would be a certificate that basically certifies that the THC is within the legal limit. And THC and CBD are linked in production. As CBD percentage increases, the THC percentage is going to increase as well within the same ratio. Most processors will not accept plants with CBD percentage less than 10% by weight. That's what they need to be economically viable. And then the ratio of THC to CBD is determined by genetics. There are plants that are CBG plants, meaning that this pathway is halted. So the plants don't go on to produce THC or CBD. The pathway stops at CBG production. And there are studies that are investigating the benefits of CBG to compare that to the benefits of CBD. So there is a problem with this arbitrary threshold, as I mentioned, this 0.3% threshold. It's very easy for growers to exceed this threshold. And since the cannabis plant produces THCA, it's the acid of THC, that's an unstable molecule. And through decarboxylation or exposure to oxygen, delta-9 THC, which is the psychoactive form of THC, is produced. So it's produced slowly over time as THCA is transformed, or it is produced immediately with heating. The federal law is moving towards a test of total THC. Right now, federal law still states that less than 0.3% THC is the limit. Some states have moved already towards setting this as the total THC threshold. So this is already a problem because about 85% of what is labeled as hemp right now, looking at delta-9 concentrations, that already would not meet federal guidelines. So if you increase this and you set that to a total THC threshold, that means that even less of those producers that have produced a crop would be able to sell their product. And that would lead to either remediation, which would include adding more stem and leaf material to the product to dilute the THC concentration, or the farmer would have to just destroy the crop that they produced, and it would be a total loss. So there is also a commodity type that is produced for oilseed, and this can be produced for both the seed and the stalks. The stalks would be a secondary commodity type, so the primary growth would be for seeds, and then the farmer could come back and harvest the stalks for fiber. But those stalks are going to be lower quality fiber than if you produce this plant that's cultivated for fiber production. These would be tall and moderately branched plants at medium density for dual purpose production. And you'd produce it for seed. Now seed begins to shatter from the bottom of the plant up. So at any time, you kind of have to gauge the maturity of the seed and look to see when you can harvest without losing too much seed from that inflorescence. There is one seed processor nearby called Victory Hemp. And we are SIU is part of this project, this S1084 federal multi-state project that is funded by the USDA. And as part of that, we conduct variety trials for fiber and seed production. So we have dual purpose cultivars that we plant across a wide geography to look and see which cultivars are best adapted to our different climates. So here we're planting some of our research in the field. We have this modified cone planter that we use for our small plot research. This was our very first year of production, and the number of insects, pollinators, and flies that were present in the field was very impressive. So this has the potential to support a diverse ecosystem as well, it appears. And this was our first planting of the Chinese cultivar Gen Ma. And there we're trying to assess fiber quality and just kind of looking at the plants. So we're looking to see which varieties were performing best or have performed best across multiple years. We're looking at differences in establishment, growth, competition, seed yield, and fiber production. And then we're still tracking our production challenges, and still we haven't worked out the details of a mechanized harvest, and I think we need specialized equipment to harvest. So looking at the fiber plants in particular, these are very tall, unbranched plants that are planted at a very high density. This would be what a fiber stand might look like. This was fiber production in Canada, and this is actually a triple crop where you can harvest seed, fiber, and essential oils. And that's what the fiber looks like when it's separated from the stem. So plants would be harvested at early flowering before seeds are set. And you may have to move through the field very slowly because the fiber may wrap around all the moving parts of the combine. So we've used a sickle bar mower in the past for our small acreages, and large acreages may need specialized equipment. This is the type of equipment that they're running in the state next to us in Kentucky. They have a 3-tiered sickle bar mower to harvest their fiber crop. And retting, so when the crop lays in the field and the microbes begin to loosen the fiber from the stalk, this has been cited as the process with the greatest need for research and standardization. So ideally, there would be some sort of maybe microbial mixture that could be sprayed across this crop to improve uniform retting. Right now, you would need to go in and flip that biomass so that it rets the product uniformly. That's right. So I have a comment, no more than 0.3% THC. Yes, I had a typo in a previous slide. Okay, so this would be a decorticator, which would separate the fiber from the stem. And then this would be how the stem is composed. So you have the hurd that is the center or the woody part, the core of that stem. It may be hollow depending on the size of the stem. And there are primary bast fiber bundles, which would be the longest usable textile fiber, or the secondary bast fiber bundles that are shorter and usable textile fibers. And from the hurd, you can create hempcrete, bedding materials, absorbents. These would be, and also particle board. For the bast fiber, you can produce plastic composites, textiles, insulation, or high-grade paper. So within all geographies, one thing that we've learned working with hemp, we need well-bred, locally adapted cultivars. So there's often, depending on and they're not true cultivars, depending on the accession that we use, we may see segregation in traits. So the plants look differently morphologically, even though they're from the same accession type. So that signifies that we need greater or better bred, like true cultivars for cultivation. And for Southern Illinois, we have a particular challenge because we have very wet springs, and we need a photoperiod insensitive cultivar so that we can plant it late after the rainfall, the heavy rainfalls occur, and it can still have time to mature before the fall season. So we need rapidly maturing cultivars that will tolerate a late planting. So there's a lot of work for plant breeders to do to help us solve some of the issues that we have. And I mentioned rainfall, so you can see the hemp here that was already established, and then we received several inches of rainfall, and you can see that the plants are now water stressed, and we lost 85% of the stand after this rainfall event. And it's because our soils are fairly heavy. This would be the soil type that I'm working in, so it's within the silt loam classification. The dots that you see here across the triangle, those are the dots where those historical victory hemp populations were found in Illinois. So you can see the type of soils that hemp, you could say, likes, where it's typically grown or where it has survived over all these decades. And then here we are on the edge of this soil type. So again, I think this is just an issue of breeding. We could find cultivars that still survive very well even in our heavy soils. So opportunities. So again, I'm a weed ecologist, so I'm interested in hemp because, first of all, weeds can be a major constraint to crop production. There's going to be competition with weeds, and we've seen that has in effect it's destroyed some of our research plots because when the weeds emerge first, they preempt the hemp and we don't get a good stand established. We find that this happens more likely when we put nitrogen down first, and then the weeds are able to germinate first and take advantage of the high nitrogen. When we delay our nitrogen application, that gives the hemp a chance to come up, and then we can apply nitrogen after the plants have emerged. So we know that weeds take up space, light, and nutrients and typically reduce yields, but it's been really hard to quantify the yield loss that weeds contribute to in a hemp stand. And there's a call for weed research in industrial hemp. So there's not really a lot known right now about how the weeds compete with the crop on a large scale. So what is the effect of weed competition on hemp yield and secondary metabolites? That's a big focus of our research. And then what is the effect of hemp secondary compounds on weed growth? So ultimately, we want to figure out how hemp can be used to suppress weeds in an agroecosystem. So that's the focus of my research. Herbicides are used across 95% of the planted acreage in the US. Specifically in Illinois, the farm size is about 151 hectares on average, but 33% of those farms are greater than 800 hectares. So our growers are trying to move across large acreages, and they're heavily reliant upon herbicides to help them with their weed control. And that puts a large amount of selection pressure on these weed populations that leads to a selection for herbicide resistance. So there are 515 cases of herbicide resistance globally. 128 in the US, which is 25% of that total. And we have more cases of herbicide resistance than any other country, which is not something to be proud of. So the main species that I work with are these species. They're considered the weeds that drive all management because they have become so difficult to control. So that is Amaranthus tuberculatus and Amaranthus palmeri. They're now resistant to almost all commonly used herbicides in our production systems. They're dioecious, so we have male and female plants within the field that have to cross-pollinate to produce seeds. They grow quickly and produce maybe up to 500,000 seeds. So this is a strong driver of evolution. And we also have hemp as a dioecious species, and we're introducing more and more cultivars of hemp into our cropping regions. So there's a good chance that we're going to see strong selection within hemp as well. And we know that it can become weedy. So in the future, it may be competing with our other crops in the fields where we plant it. And we may be pushing it as far as evolution and even selection for herbicide resistance in the future, once we have enough acreage planted of hemp. So I'm focused on integrated weed management. I would like to see our US cropping systems move beyond just herbicides and integrate more of these types of cultural and mechanical tactics into our production systems. So cannabis or hemp fits within this framework because it could help us diversify our rotations. It could expand that very simplified corn and soybean rotation. So I've done some work looking at competition and specifically and the effect on hemp yield and secondary metabolites. So we've looked at a CBD type cannabis, and we know that cannabis is very rich in phytochemistry, and these secondary metabolites are often used to attract pollinators, to defend against herbivores, and in other plant-to-plant interactions. So hemp has about 2% of total dry mass that can be cannabinoids, and 20% of the inflorescence can be composed of cannabinoids. And it produces dozens of terpenes that are semi-volatile compounds that impart flavor and scent, such as limonene here as one of the terpenes. So we have investigated competition in plasticulture because, again, that's a very common production practice. So we put Amaranthus tuberculatus at different densities in competition with hemp plants within the planting holes. Hemp was transplanted and water hemp was directly seeded into these systems. And we found no reduction in biomass for the hemp, no matter how many plants of water hemp or Amaranthus were competing. And we found no differences in cannabinoid or terpene content by treatment either. We did see differences by year. So we know that there's an environmental influence on the production of terpenes, but it doesn't seem to be associated with competition. And then we've done some work of similar type in our fiber and dual purpose cultivars. We planted these late again, so you'll notice this was past the summer solstice, but we still were able to achieve a high biomass crop, and we harvested on September 22nd. So this was a late planting, but it seems to work for us when we have the right cultivar. We weeded these plots up to 87 days. So there were some plots we didn't weed at all. Some we weeded for 7 days, 14 days, 28 days, and so on. And it was truly amazing that we saw no difference. It didn't matter if we didn't weed the plots at all, or if we weeded the plots for 87 days, the biomass that was produced was the same. And this is counterintuitive to what I think I see. So we're trying to dig in a little bit more and try to explain this and really quantify the competitive effect of weeds on the plant. We did have an average of about 10 tons of biomass per hectare for this one Chinese cultivar, Genma, but we also had Joey, which did not do well in our geography, and then here this Northwest Genetics 2730, which did fairly well. These were dual purpose cultivars, so it's not quite a fair comparison to this fiber only cultivar that would produce high amounts of biomass. So the effect of hemp secondary metabolites on weed growth, it's very possible that some of the secondary metabolites in hemp can be harnessed and used for weed suppression in the future. And we investigated this by looking at hemp residue. People have looked at individual compounds and their effect on weed growth and the growth and germination of other plants. but no one, as far as I found, had looked at the whole residue before. So we applied the whole residue to the soil surface. We placed seeds below that residue where the seeds had good seed to soil contact, and our control residue treatment was a maize treatment. So we assigned pots to 3 groups where we had soil alone with no residue, 5 grams of hemp residue on the surface, or 5 grams of maize residue on the surface. And maize was included just to control for that mulch effect. And seeds were allowed to germinate over 30 days and misted daily. And seedlings were removed as they emerged. And we used this time to germination test to look at differences in emergence. So what we found was that germination was dependent upon the environment that we'd created and the residue type. We had many fewer seeds that you see here in the purple that germinated in our hemp residue treatments as compared to the maize in orange and the soil alone in green. And these were, again, amaranthus seeds. So there's a new type of weed control practice that is gaining traction in the U.S. We've basically imported this from Australian weed scientists and farmers, where it's the practice of harvest weed seed control, where you manage the soil seed bank by controlling the seeds at harvest. You destroy the seeds either through here you'll see an integrated seed destructor within the combine. So the seeds are being pulverized and then pushed back out the back of the combine. So it devitalizes the seed and controls the seed bank. There is also another type of practice called chaff lining, where the seeds aren't destroyed. It's much cheaper for growers to do this because they don't have to have specialized, expensive equipment. They can just build a chute to go on the back of their combine to funnel the finest particulates of the crop and the weed into one space. And here's an example of a chaff line in Australia. The weeds emerge in this line. And over time, if you do this practice over and over, you can concentrate the weeds in these lines. Australian farmers with integrated livestock systems graze these lines early in the season. There's also the possibility to broadcast a non-selective herbicide to control the weeds in these lines and not harm the crop in the other parts of the field. So with this new tactic, this makes the allelopathic compounds of hemp relevant. So we could potentially have a hemp production system where at harvest, we come in and we harvest the hemp crop, we funnel all the residue out the back of the combine, and any weed seeds sit within this allelopathic residue that's going to further inhibit their germination. So it's possible that we can use the hemp residue within our corn and soybean rotations to help suppress weeds and form a new type of weed control tactic in the US. But in order to do that, there are some limitations, right? We have to figure out the persistence and the availability of these chemicals throughout the year. And most importantly, we have to have a market for hemp that continues to develop in the US so that it's profitable for farmers to do this within their rotations. So with that, I think I've gone a little over time, but I thank you very much for being here with me today, and I'd like to take any questions.
Thank you, Dr. Gage, for your clear and very interesting presentation. I ask the audience if anyone has a question. Maybe you can raise your hand or write them up on the chat dashboard. I think we have a couple comments or maybe questions that we can go through, waiting for other questions to come up. Well, the first comments are just greetings from people. And then we have a comment from Zakaria Kamal saying that I'm a Moroccan researcher scientist. My topic is studying essential oils of cannabis and its different use on medicinal and therapeutical cosmetic levels. I would like to join the LinkedIn network of Dr. KARLA GAGE so we can discuss. But so he's just trying to join your LinkedIn webpage maybe.
And please, I would say, please also send me an email. You know, I'm not as good with my social media as I should be. So please contact me directly through my email.
Okay, so Zakaria, please contact Dr. Gage by email if you want to join in her LinkedIn webpage. And I think there is another comment that you already addressed during your presentation regarding the percentage of the THC. So that it was a typo on your presentation, 0.3. 0.03% instead of 0.3%. So it's already, you've already addressed this. And I think there is another question from Barada Abdel Fattah saying, do you recommend using hemp as a cover crop?
So there are researchers that are interested in doing this. There are some limitations right now because hemp seed is still very expensive to use as a cover crop that you don't harvest, right? So just simply to suppress weeds, in the future, it might be possible to, harvest this as forage. And right now you would not be legally allowed to feed hemp to livestock, but I could see that becoming possible in the future. But, it would be a challenge looking at the way that our crops are planted because hemp is not going to germinate until, it can germinate in very cold conditions depending on the cultivar or the accession that you have. But it wouldn't germinate until, here early or mid-March at the earliest, likely. And then you would want to go in, early May. That's when most of our producers are trying to plant their hemp, their soybean or their corn crops. So that's not very much time for the hemp to grow and produce biomass. It does grow quickly, so maybe it would be an effective cover crop. It's something that needs further research, and I don't think it's going to be possible until we have a cheap source of seed to plant for that. So it's possible, but at this time, it's not really feasible.
Okay. Well, I think waiting for more questions from people. I have myself a couple questions. You talked about in your presentation about the THC threshold, that which determines whether the production can be commercialized or not. My question is that are there any studies going on to select for plant that maybe produce that percentage within the legal range? Some kind of research or breeding studies going on there?
Yes, that's a huge line of research right now, and the state governments collect information on the cultivars that are planted in their states, and then they take note of the ones that exceed that THC threshold. Some states go so far as to remove those from the list of possible cultivars that their growers can plant. So it's very important for hemp breeders and retailers and dealers that sell those seeds. They often give growers recommendations on when to harvest that crop to prevent exceeding that 0.3% threshold. Because if the grower exceeds that threshold and then reports that back to the state, depending on the state, that cultivar could be somewhat, I guess you could say, banned from production within that state. Now, Illinois doesn't ban cultivars. I don't have a list of things that I can't plant, but I just have to deal with the consequences if I go beyond that 0.3%. And it's a little different for us for research. All the things that I've grown over the years, I just destroy those products, right? I don't put anything into commerce from research. And the state doesn't require that I test everything for THC. I can grow the plants, take my data, and then destroy the biomass, and that's okay. But for a grower, it's much more restrictive.
Here. This is just a reminder to the audience that you can ask questions in English or maybe in French, and we can try to translate to Dr. Gage. So please feel free to ask the question the way you want. May I ask another question? So it's about, I'm gonna just put my plant pathologist hat, and I'm just curious to know whether there are some significant diseases that proliferate on a weed or on a hemp plot. Maybe some very important disease that can be, or maybe pests as well.
Yes, so that is not my field of study, but I have observed fungal pathogens on our plants. You know, I had what I thought was botrytis, but in talking with plant pathologists, it was likely a species of Fusarium instead. But, you know, I've seen that. I've seen, I believe, what is Septoria, and maybe even some Cercospora on the leaves in our fields. There's also some root fungal pathogens that can infect the crop. There's one called southern blight, and I can't remember the genus even right now. But as we plant more acreage of hemp, we're probably going to see more of these pathogens evolve. And there's a big question right now as far as, you know, I'm talking about rotations and putting this in rotation with corn and soybean crops, but we don't truly know what pathogens that these crops may share in common. And that's, you know, plant pathologists are working on that. Nicole Gaucher from the University of Kentucky was also at the Amols conference, and she spoke about her work investigating fungal pathogens. And she's been developing a database of pathogens that she's found on hemp and working with growers. As far as insects, we do have a number of lepidopterans that come in at harvest time. They seem to be highly attracted to the floral biomass. The mode of attraction, as far as I know, is unclear right now as far as what signals the plant is using, or, you know, how the lepidopterans are finding the hemp plants. But that can significantly decrease the floral quality, right? So if you have lepidopterans that are boring into the inflorescence and leaving waste behind, that's going to be a low quality product and you may not even be able to sell it. There are very few products that can be used to control lepidopterans. And they're all INRA certified organic compounds. But once the lepidopterans begin to bore into the inflorescence, there's really nothing that you can do because the contact between the product and the insect it's not going to be good.
Thank you. I think we have Azim Khalid who would like to ask a question. Dr. Azim?
Yes, hello everyone. Thank you, Dr. Gage, for your nice intervention. I'm a researcher in organic agriculture and the optimization of organic waste management, and my questions will be on those two aspects. The first one, is there any market for organic hemp in the United States or worldwide? I'm not a researcher in this field, but maybe you can have some information about this. This is my first question. And the second one, you said in your last comment that you get rid of the biomass after your researches. Why not using those biomass into composting process for two reasons? The first one is to make the biomass in circular economy and turn it around and get an organic amendment to be restituted to the soil. First of all, and second is to try to find out how allelopathic effects of organic biomass of hemp would deal with the seed weed problem during the composting process. That would be a nice topic for research. Thank you.
I agree. Yes, that's an excellent topic for research. You know, as far as how we discard our biomass, in the past, we've destroyed it by burning. The state tells us the ways that we can destroy it legally, and we can burn it, we can bury it, we can compost it. We are looking at a compost facility right now to try to determine if the compost facility reaches temperatures that are going to destroy the seeds, which is our main consideration. We don't want to spread the seeds. But composting the biomass makes a lot of sense, right? I mean, why destroy something completely when you can use it? And I agree that the question of whether those allelopathic compounds persist in compost would be that would be really good to know, right? And it could be even more useful as a compost additive if it further suppresses germination of seeds in compost. So I think that's a great line of research. As far as the market for organic hemp, right now, as far as I understand, the seed market is still primarily an organic market. And, you know, most of our corn and soybean growers, they are heavily herbicide dependent, right? So they're not organic certified. So anything that they plant is not going to be organic, even though they're not allowed to use products on it while they're cultivating it. So right now there are no herbicides that are federally approved to use in a hemp crop. We're going to see that change over the next year, I would predict, and states have already moved forward. Some states allow, for example, there's a grass-specific compound that you can use post-emergence to at least take out the grasses, but not the broadleaves. And then there's a pre-emergence herbicide that you can use to help, give the hemp a head start and hold back the weeds from germinating. So right now, yes, primarily an organic market for seed. And if you're growing anything for CBD extraction, it's a very important issue. So those farmers can't they can use so few compounds, and all of those are the INRA certified organic compounds, but there's a very small list of products that they're able to use. So that is only, I should say, an organic market. And that is because when you think about using the floral biomass and you're going to concentrate it, you're going to extract the CBD, if there's any toxin, anything that's been applied to that product is just going to be concentrated in that CBD production. So it would be counterproductive, right? If you're going to use that as a medicinal crop, and any sort of health benefit if you're concentrating toxins in the crop. So
thank you very much.
Yes, you're welcome.
Dr. Sarri, with your mic, please.
Oh, sorry. We've got another question from Dr. Boulala Mohammed. Dr. Boulala, go ahead, please.
Oh, thank you for this very interesting presentation. My question will be more in economic aspects, and I hope that you have answer. So yeah, as you mentioned in your presentation, profitability is a key driver for farmers to grow hemp, and it will be the same in Morocco. Do you have an idea about the profitability per hectare, for example, in your area, or just a comparison between the profitability for hemp or
Okay, there. That is pretty far out of my research area, but let me at least tell you some observations that I've made. So in 2017, while university researchers were the only ones that could really cultivate hemp on a large scale, that was when the supply was really, really low for CBD production. And there was a grower in Missouri that wanted to work with my university, and he was claiming that he could get $75,000 an acre for hemp for CBD production. The following year, it was down to about half of that, 35,000. And then as more people started growing it, the market just bottomed out. And then growers, in some cases, their contracts with processors were not honored, so they were left with product that they couldn't sell. And some growers have been storing product for 2 years now, and there's no way. I mean, there's no way they can sell it because the quality, I'm sure, has decreased to a point that there's not a lot they can do with it. So it was a market that kind of went boom and bust, basically. So there's still a lot of products that are using CBD, and there's still demand, but the supply has exceeded the demand. And I'm not sure currently this year where that stands. So if you look at the other markets, right? So Canada supplies a lot of the seed for the products that are created, but there's believe, and again, not being my area of expertise, I believe that there's room to expand that seed market depending on the products that are created. And then the fiber market is where I'm primarily concerned, because I see that as that's where we're going to improve our agronomic production of those other crops, right? By growing a fiber crop that's going to suppress weeds. So I would really like to see that grow and expand in Illinois. We have 2 fiber processors that are relatively close to our region in Southern Illinois. But in order for fiber to be profitable for a grower, there really needs to be a processor within about 50 miles, so there's a radius where you're gonna lose money in transport, because you have to have such a large quantity of biomass that you have to ship to a processor. So it's you have to have processors that are located near a group of growers that are going to provide enough biomass to supply that processor year round. And I've seen some numbers, but I can't right now. I'm afraid if I quoted anything, I would get it wrong as far as the number of tons per acre that would need to be produced for each grower, and the number of acres that it would take for a facility on average to operate year round. So there's that type of market analysis is being done, and there are a lot of people interested in increasing the amount of fiber production and processors. But the processors aren't going to emerge until there are growers that are able to grow hemp, right? And we still have a number of agronomic challenges. We're still working out the best management practices for growing hemp for fiber. So as we gain more knowledge and we understand how to produce a better crop, and growers become more comfortable, I think we'll see more people invest in processing facilities. And I've heard that on average, it's going to cost $10 million or more to create a processing facility.
Thank you.
Thank you. I think we've got another question posted on the chat dashboard. I'm going to just try to rephrase it depending on my understanding of that question. So Zachariah asks whether cannabis competing with other crops is a topic that is not covered enough yet.
I haven't seen the research. So in my opinion, yes. We, in our own research, we see if the hemp is slow to emerge, the weeds emerge first, and we don't get a crop, right? And then if the hemp emerges, it seems to be very competitive with other plants. So I would assume, we're establishing a seed bank. Whenever we plant hemp in a field, not all of those seeds are going to germinate. So we're going to have what we call volunteers the next season. So all these hemp plants that are going to come out of the soil and compete with soybeans and corn. So hemp may be a highly competitive weed in these other systems. But right now, as long as you're not an organic producer, there are a lot of herbicides that will control hemp in other crops. So I don't think it's going to be a problem initially, but I mentioned the aspect of dioecy and how the plants have to exchange genetic material in order to produce seeds. So the evolutionary potential of hemp as a weed to evolve to be an even better competitor in these other cropping systems, that's going to be I mean, I could probably focus my lab's research only on that in the future because there are so many questions there.
Okay, talking about hemp competitiveness, I have another question. What are your thoughts about the insignificant difference between the weeded and the unweeded plot that you presented in your talk? Do you have any hypothesis, the first idea, the first shot?
Well, let me go back, and I want to show you one thing that I didn't point out because I was afraid I didn't have enough time. Okay, so you see this this 14-day biomass here. When I saw the data, I knew something had happened, and I went and talked to my field crew, my students, and my researchers that were working on this project, and they were delayed in removing the grass competition. We were using a post-emergence application of a grass-specific herbicide. So I can see a signature of grass competition in my study. So I think, you know, not all weeds are going to compete with hemp equally. We have a lot of problems with dominant grasses in our system, but we can remove them with grass-specific herbicides. But when we don't do that, we see the competition effect. I guess, you know, that call for research that I mentioned in the literature, you know, that was put out. Where am I going? There. That was actually put out because they did a review of all publications that, you know, discussed hemp production or cannabis production. And they found that most of them didn't mention the weeds at all. So they had no mention of weed control. And that was, you know, a clear indication that hemp may be a very strong competitor. You'll have some people that say that hemp doesn't need herbicides, it doesn't need any weed control. I don't think that's true because there are going to be a lot of cases where you can't get the hemp established fast enough. We'll have weeds that establish faster. But it's a time issue, a temporal issue. If the hemp crop emerges first, it can outcompete most other weeds, I believe. So I think that's what happened. You know, in our plots where we didn't do any weeding at all, we just saw the hemp emerge, and it was competitive against those other species. You know, so that's what we were looking at. Now I'm working with another researcher at SIU that is planning to look at species of concern in agricultural systems. And we're looking at doing a couple of different types of competition studies in pots in the greenhouse to kind of try to remove all of these external factors in the field and really get a good understanding of that individual plant-to-plant interaction and how competition affects the growth of hemp and how hemp suppresses those other weeds. So it's something that we weren't able to really pick up in our field production. It looks promising, right, for growers if they can figure out how to get the hemp crop established well early.
Thank you. Well, probably we're going to take a last question that is posted on the chat dashboard. So Barada Abdel Fattah asks, did you look at the correlation between hemp plant density and weed competition?
No, we have the data, and yes, I will see you in Fort Collins. I'm about to leave to go to that meeting about hemp, actually. But we're gonna that competition study that I was discussing in pots, that will be a plant density study. So we'll be able to tease that out in the greenhouse. In the field, we have the data. My goal with the data that we collected is to run a plant community analysis. So a non-metric, multidimensional scaling approach so that we can look at differences in the communities of weeds associated with the hemp that we planted. And as part of this USDA study that I'm showing here, the graphs from, we have different planting densities of hemp. So we can look at densities of hemp and how they affect the densities of the weeds within that community and the species identity as well. So for this weeding study here, you know, I can look at the change in the community over time. And this is relevant from an issue of a post-emergence herbicide application. We were trying to find the point at which you would have to do you know, when you should time your weed control measures for the greatest profitability. So we were looking for the critical weed-free period, how long you have to control the weeds in order to get a good crop yield, to try to time that post-emergence herbicide application.
Okay, thank you. Well, if there are no more questions, I would just like to mention that there is a survey link on the chat dashboard for people who would like to provide assessment guidance to the organizers of this seminar or of this webinar. It will be very helpful for future improvements. You can also request your certificate of attendance through this link. Well, finally, speaking for myself and on behalf of the organizers, I'd like to thank very much our guest speakers, Dr. KARLA GAGE, for her time, availability in the preparation of today's webinar and for sharing with us her knowledge, Many thanks also go to all the participants and extend especially to my colleagues Boutaina Abidou, Uthman Sbatar, Ardad Tirazi, Nordin Bahri, and Abdelaziz Yasri, and all the scientific committee of the INRA webinar series for their crucial contribution in the organization of this series. Dr. Gage, do you like to say something at the end of this session?
Yes, I would like to extend my thanks to INRA. Thank you so much for this invitation. And please, if you have additional questions, just please reach out. I would love to build more collaborations with Moroccan researchers as you learn how this crop fits into your production systems.
Thank you. So finally, just an announcement. The next webinar in this mini series is scheduled on March the 2nd. Information on this sixth webinar is showing up on the screen. It will be given by Dr. Josie Saladin, who will talk about genomics of cannabis and breeding. So hope to see you there next month. And thank you, everyone. Bye.
Thank you.
Thank you, and bye-bye.